All right. Hi, everyone, and welcome. This is Caroline with Fiveable, and I am so happy that you tuned in tonight because we have a big night ahead of us. We're going to be talking about chi-square analysis, which is always relatively tricky. I'm realizing that my glasses are giving me really weird glare. But chi-square is oftentimes really tricky. I haven't seen it on a free response in a few years, which is always kind of a worry, I suppose, because it means maybe this year is the year. It's hard to say, but I am going to be working through my PowerPoint. Let me think. Give me just a second. So I made instead of Google Slides this week to kind of make life a little bit easier. So if you tuned in last week when we were talking about dihybrid crosses and things like that, it was hard for me to like switch back and forth between a couple different entities to write things out. So this time I made a regular PowerPoint because it's a lot easier to write on those. So give me just a second. Let me see if I can still share this link with you because I did upload it into the drive. And it would be nice for you all to have a copy out in front of you. Otherwise, you'll just have to look at my screen. No big deal. So let me find that. Goodness, why is this taking so long? Also, usually I do my casts at school, but I'm obviously at home right now, and for some reason my dog just knows. She knows when I am not paying attention to her and doing something else important, and just decides to be super annoying. So if you hear her, that's my dog. All right, let's see. It looks like I can open this up with Google Slides. So I'll send you that link, and then I'm going to share you my screen on a regular PowerPoint whenever this decides to open. Okay, cool. Here you go. And I will share my PowerPoint with you. I cruise in here because this is a big topic. If you have gone over this in class before, then you know that it's a challenging topic. If you have not, then this is great that you're tuning in because your life is going to be a whole lot easier when everyone else is really stressed about chi-square. Okay, here we go. All right, so we're going to hop in. So as I said, my name is Caroline. This is Fiveable. We're talking chi-square analysis. So the first thing to understand here, and the reason why we're going to talk about this now in the middle of kind of talking about Mendelian and soon non-Mendelian genetics, is because chi-square is a test that allows us to tell if variation in data is due to chance or if it's due to a variable that we're testing or some other influencer on a situation. So in plain words, chi-square is a way to determine if your data is legit or if a situation is legitimately just due to the random nature of the universe or if it's due to some other power that's influencing, like we could use this to determine if genes are controlled regularly or if there's some sort of like another linked gene that's having an effect on the genes that we're seeing, anything like that. So we're going to use this today primarily to look at genetics problems and see if results that we see in real life match up with what we would expect from a Punnett square or something like that. So we can use this in order to compare if our data is close enough to what we would expect, that we could say, yep, this is just due to chance and the random nature of the universe, or it's far enough away that we're like, this is actually really significant. Something else is at play here. Maybe it's a variable that we expected. Maybe it's not. Regardless, we're going to take a deeper dive. Okay. All right. So there are some equations that we're going to be using. They're really easy, actually. I know it looks kind of scary. We have this little alligator mouth. That's actually a sigma, and it means the sum of a lot of different points, and I'll show you what that means once we start manipulating the data here. All of this is actually exactly taken from your equation sheet that you get on the AP exam. So this is all given to you. None of it needs to be memorized, and we will work through every part here over the next few slides so that you feel really comfortable with all of this data, and then it's there for you. You don't need to remember any of it. You just need to remember how to apply it, okay? So all of your questions about these formulas, this table, all of it will be answered in the next few slides, and then we'll do some practice. So if you're confused at first when I'm just going through this verbally and showing you what the key terms mean, just sit tight because once we start doing practice, everything's going to make a whole lot more sense. Okay, so some important vocab to know. So the chi-square test I already talked about, but this is a test to determine whether results are significantly different from some sort of expected value. Where that expected value comes from can change depending on the situation. For today, it will always be from a Punnett square test. So that's where we'll get what we expect, and then we'll compare what we actually see with those expected values. We're also going to talk about something called a null hypothesis. And null is oftentimes used in math, so maybe you've heard it in your math classes before, but it kind of means none or nothing. So a null hypothesis is a hypothesis that assumes that the results or outcomes that we see are due to chance. So it's going to be our job then to either accept or reject or fail to reject, depending on how your teacher describes it. It used to be like a really big deal that you didn't say that you ever accepted the null. It was either reject or fail to reject, but AP doesn't care anymore. So depending on your teacher, either of those languages will be used. But this null hypothesis is basically what we are putting up against to see if we agree with or don't agree with the statement that our results or the outcomes that we see in a certain situation are due to chance. Okay. The next important thing that that we need to be familiar with is something called degrees of freedom. Okay, degrees of freedom is a term that is actually given to you. So if we flip back a few slides, this is actually described. But degrees of freedom are equal to the number of distinct possible outcomes in a situation minus 1. Okay, so it's the number of possible outcomes in a situation minus 1. So if a dog can have spots or stripes, then the number of degrees of freedom for that would be 2. Or sorry, 1. Oh my gosh, there's 2 possible outcomes, spots or stripes, minus 1. So we would have 1 degree of freedom for that specific situation. If it could have stripes, spots, or have plain fur, right, then we would have 3 possible outcomes minus 1 or 2 degrees of freedom in that situation. And this will make more sense when we start using actual scenarios, and you'll see like, oh, here's the number of possible phenotypic outcomes, and we'll subtract 1 from that number, and that will give us our degree of freedom, which we are then going to use in order to determine our critical value, okay? So our critical value is listed in that chi-square table. That's a few slides back. I can flip back to it in a second. But it's just a measure of comparison. Okay, so we'll use our degrees of freedom to find our critical value from the chi-square table. And we'll use that value and compare it to the chi-square value that we calculate. Sorry, my dog is doing something out there. She's a mess. Sorry, so we'll compare that to a value that we calculate given the formula that was on a few slides ago. So let me go back here for a second. So let me get my pointer out. So these are our critical values in this table. So you'll see at the top here, we have our degrees of freedom. Okay, so in the scenario that we were talking about, if we had a dog that could have spots or stripes with 1 degree of freedom, we would be looking at this column of our chi-squared table. And you'll notice that there's 2 numbers that live in this column. These are the 2 critical values that you would focus on with 1 degree of freedom. We'll get into a little bit, I don't want to get into it right yet, but once we do our first practice problem, I'll tell you a little bit more about what these p-values mean. You're always going to use a p-value of 0.05, but I'll explain that a little bit more when it becomes more relevant in just a few minutes. Okay. Sorry, I think my dog has my shoes. I will be right back, but read through the purpose, and then when I get back, I will explain to you what it means, because it's very important, and I have a creative little analogy that I came up with. Remember it. And we're back. Okay, so our purpose here, once we have our critical value, our degrees of freedom, we have our null hypothesis, we're trying to either accept or reject it. We have the ending purpose then. So if our value, right, the value that we have calculated, okay, using the equation is greater than or bigger than the critical value that we're comparing it to from the table, then our null hypothesis is rejected, okay? And if you look back up at our null, the null is a hypothesis that assumes results or outcomes are due to chance. So if we reject that, it means that the results are not due to chance, right? They're significantly different, and we can prove that. So if you become a scientist one day, this test actually becomes incredibly important for you to tell the rest of the scientific community that your results are significant and not just due to the chance nature of the universe. You're like, no, this is legit. These results, these differences that you're seeing are actually significant enough that we know that it's because of this variable that we expected and not because of just chance. Okay, so if the value that we calculate, and again, once we start doing practice, this will become a little bit simpler, but if the value that we calculate based on our data is greater than the critical value, then our null is rejected, the results are not due to chance, okay? They are significant. The way I tell my students to remember this is very strange, but it works. And honestly, this purpose portion is the only thing that you need to remember about chi-square, because everything else is given to you, right? The equation's there, your critical values are there, the definition of degrees of freedom is there. You have to remember the purpose, okay? So this is what I tell them. If you see a very small number, let's say 1, you see one person running down the street, naked maybe, covered in feathers, glitter, you're like, wow, that person's crazy. Okay, this is such a chance event that I have stumbled across this crazy person, just one of them, small number of them, that's doing something crazy. Okay, so if your value is smaller, if it's a small number than your critical value, that's just due to chance. Just like that crazy dude running down the street, it's just due to chance that you ran into him. If instead 50 people are running down the street naked, covered in glitter and feathers, right? 1 might need to get inside because something crazy is happening. But 2, that large number of people doing something crazy is probably not due to chance. It's probably due to the fact that they're insane and they're in some kind of crazy organization together and they planned to go all do this crazy thing on this day. Okay, so if you have a very large number, right, then you're like, okay, this is not due to chance. Something crazy is happening here. There's something else affecting this situation. So I know that's insane, but sometimes insane things help people remember things. So maybe that'll stick with you. The other thing to mention here, right, if you think about it, if you're more like, hey, Caroline, that's nuts. I don't work that way. That's how my brain works. So there's a scary insight into me as a person. But the other way to think about this, if you have a really big number, our numerator here is just the difference between what we observe and what we expected squared. So if you have a really big number, it means you have a really big difference between what you observed and what you expected to observe. So it should make sense that a larger chi-squared value means this is not due to chance because there was a larger than expected difference between your observed and expected variables. Okay? Okay. Now I haven't looked at you in a while because I'm using PowerPoint, so I can't just like flip back and see the conversation. So let me pop back and just make sure we're all good. Everyone followed me so far? I don't see any questions, which is great. Maybe they'll come when we start doing practice. Hopefully you don't mind my rantings about people running down the street covered in glitter. Okay. There's my dog. So she's decided to now join us. Want to see her? She's very cute. She's also very bad. Okay. All right. So back into it. Hi, Addie. This is why you should never cast from home. Okay, and we're back. All right, so we're going to do some practice problems now. I'm going to walk you through every single step of these practice problems. It's going to seem a little redundant, but I can promise you it will make sure that you understand the underlying concepts so that you don't make silly mistakes if you see this problem again, okay? I'm going to use my handy dandy pen. For those of you who tune in frequently, you're going to realize that every week I'm getting better and better with my technology, okay? So I'm going to use this. We'll write out a few things. We'll walk through a few steps, and hopefully by the end, you have a better understanding. Okay, so our question. If a black heterozygous guinea pig is crossed with a homozygous white guinea pig with the genotype b, what is the probability that the offspring will be white? Okay, so this was a problem, and we actually don't need to know the probability that the offspring will be white yet, but let's just do this Punnett square for starters, because this is going to help us get our expected values, okay? So we have a heterozygous guinea pig, so that's going to be capital B, lowercase b, crossed with a homozygous white guinea pig, lowercase b, lowercase b. So if I fill this in quickly, boom, 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 boom. I really need a mouse. I guess that would make this way easier. Maybe one day, okay? And we get these results, okay? So I can see right off the bat that we have about a 50-50 chance here for offspring between this heterozygous black guinea pig and this homozygous white guinea pig. It's predicted that 50% or 2 out of 4 of their offspring would be black, and the other 50% or 2 out of 4 would be white, okay? All right, so here are the steps that we're going to walk through then in order to figure out the data that we need to go through our chi-square analysis. Okay, so first question, how many possible phenotype outcomes are there, okay? How many possible ways are there for this guinea pig to look, okay? So if you look back in the problem, this is a simple monohybrid cross of one Mendelian trait. Guinea pig can be black, guinea pig can be white. So there's 2 possible phenotype outcomes. Do not get confused thinking that there's 3 because there would be 3 possible genotypes. They could be homozygous dominant, heterozygous, or homozygous recessive. We just care about phenotypes here. So there's 2 possible ways this guinea pig can look, black or white, okay? Knowing that, it's really easy to calculate our degrees of freedom. Remember that our degrees of freedom is our possible outcomes minus 1, okay? So we have 2 possible outcomes, minus 1 will give us 1 degree of freedom. Easy enough, okay? So we have 2 possible outcomes, 1 degree of freedom because of that. All right. So now we get some actual data, and we're going to manipulate this in a second using a little table, okay? So all of that was theoretical land. We know what we would expect from this situation, okay? Now we actually see these guinea pigs. We interview them. They had a bunch of babies, 40 babies. They were very busy. And this is what we find. We find that 29 of those guinea pig babies were black, and 11 of those guinea pigs were white, okay? So while the Punnett square gave us our expected values, these values are what we actually observed, okay? So these are our observed, that looks nothing like the word observed, but these are our observed values, okay? All right, reflecting on what we found from our Punnett square, we could easily then figure out how many we would expect to be white and black if Mendelian traits were all followed, okay? So if they had 40 babies, how many would we expect to be white and black? And this is going to come, and I'll write a note here, from our Punnett square. So if you flip back a slide, you'll notice that our Punnett square predicted we would have a 50-50 split, okay? 2 out of the 4 boxes were dominant, and 2 out of the 4 boxes were recessive, okay? So my students like to set up proportions for that. You can either multiply by a fraction, you can set up a proportion, you can multiply by a decimal, right? But if we expected 2 out of 4 boxes to be dominant, how many would that be out of 40? More importantly, if you just think about it, we would expect 20 guinea pigs to be black and 20 to be white, given our Punnett square, okay? So with our expected values, again, those expected values are always going to come from some basis. You're not just going to make them up, right? And for our genetics problems, they're always going to come from the Punnett square that you conducted, okay? All right, let me just check back because I know we just did a lot. Oops. Keep those guys. Make sure we have no questions. Okay, we're still good. I'm still going to cruise then. Here we go. All right, so we have all the information that we need. Now we just need to do some relatively simple math And we can maybe disprove the null. So our null hypothesis, remember, is always that our results are due to chance. Any differences that we see are due to the random nature of mating, okay? So let's see if that's true or not, okay? So we're going to use this handy dandy table, which would usually be provided for you. So you can rewrite the null hypothesis up here, okay? And then you have your observed and your expected values, and then the equation that we're going to use, okay? So we observed, if you go back to the previous slide, this is what we actually saw when we looked at the guinea pig babies. We observed 29 black guinea pigs and 11 white guinea pigs. Okay, so our total, this is blacked out, but I actually like to write the total every time because it keeps me accountable to making sure my observed and expected are the same, because you can make some funky errors here and that will lead to some problems. But if you can double check and make sure that your observed and expected add up to the same thing, you can avoid some of those, okay? So that's what we observed. We expected 20 from the simple math that we did based on our Punnett square. This likes to crap out on you, okay? And now we need to do some math. So this right here is the formula that's given to you on the exam, and if you flip back a few slides, you'll see it's there just as it's written here, minus the little alligator mouth sigma, okay? And depending on how confident you feel with your calculator, you can break this into 3 steps or you can do it all at once, okay? But the steps are relatively simple, okay? So step 1 is just your observed minus your expected, okay? So in this case, if I was doing this for the black guinea pigs, observed 29 minus expected 20, I get 9, okay? The second step is to take that answer, okay, and square it. Easy enough. So observed minus expected was 9. If I square that, I get 81, okay? Then the last part is to divide your answer by the expected value, okay? So I had 81. If I divide that by 20, I would get 4.05 as my chi-squared value for the black guinea pigs. Now, I know this is not that challenging. It's pretty simple algebra. The reason I kind of spelled it out for you here is because I've seen a lot of student errors, like not using order of operations correctly, not putting parentheses around things. So it's like observed minus expected squared without the parentheses. So then it's like 29 minus 400 instead of 29 minus 29 squared. So 400 versus 81 is pretty substantial. I've seen a lot of weird stuff. So that's why I'm making this a big deal. Be really careful with your order of operations as you cruise through these. Okay. We need to do the same things now for the white guinea pigs. So observed minus expected, 11 minus 20, negative 9, squared, 81, divided by 20. It just so happens, and this will not always happen, but it does in this situation, that we will get the exact same answer for this one, okay? So you go through this process, right, of observed minus expected squared divided by expected for each one of your possible phenotypes. So in this situation, it's just 2. We'll do a dihybrid cross example, and that's going to be 4 phenotypic possibilities, okay? And then we end up with these chi-squared values. And where that alligator mouth comes in, right, this guy that you see in your equation sheet, is that we have to add all of these up in order to get our sum total chi-squared value, okay? So we'll add this up. When you add 4.05 and 4.05, you'll get 8.10 as your chi-squared value, okay? So this number doesn't really mean anything yet. It's important, but now we have to compare it, if you remember, to our critical values from our table, which is a few slides prior, to see if this is going to be supported or rejected, as far as if these results are due to chance or something else, maybe guinea pig infidelity, okay? So our chi-squared value is 0.10, whoops, okay? And we'll go back and look at our chi-squared table in order to find our critical value. So if we go back, oops, right, we have 1 degree of freedom in this situation, right, because we have 2 possible phenotypes. So 1 degree of freedom, we're looking at these critical values, and for all intents and purposes, unless you are told otherwise, you should be using the p-value of 0.05. So I'll briefly explain, not that you need to know, because you can just remember this. So you can tune me out if you don't actually care the reasonings why. But basically, what a p-value of 0.05 refers to is statisticians, the people that came up with these numbers, right, basically put like a stamp of approval on that number and how much they support it. Okay, so in other words, if our chi-square value is greater than this critical value, with the p-value 0.05, we are 95% sure that the assumption that we make is correct. Okay, we're 95% confident that yes, these results are due to chance, or yes, these results are not due to chance. Okay, if you use the p-value of 0.01, you'll notice those critical values are higher. It's going to be harder for our chi-squared value to be greater than them. Then you're 99% confident in your assumption or whatever conclusion you draw from those results. Okay, so it's a strange process. Statisticians are crazy people, right? But for all intents and purposes, you just need to know that the p-value of 0.05 is good enough. We are good enough with that A. We are good enough with a 95% confidence in our answer choice. Okay, so knowing those two things, we will find the critical value that we are comparing our chi-square value to is 3.84. 1 degree of freedom, p-value of 0.05. We have a, once I can get to it, a critical value, which I'm going to abbreviate as CV, equal to 3.84. Okay? So in comparing these two numbers, what you will notice is our chi-square value is greater than, you can see I'm having like a lot of fun with colors here. I usually use lots of colorful pens in my classroom. So our chi-square value is greater than our critical value. And if you go back a few slides to where we talked about the purpose of this whole thing, that means when you have a large number, just like a lot of crazy people running down the street, that we can reject the null. Okay, these results are too far from what we expected. They are not due to chance. Something else is going on here. So I'm going to add that to the slide. We are going to go ahead and what color? Reject the null. Ooh, fancy cursive there. Yikes. Reject null, which means something is going on with these guinea pigs because it's not genetics. It's not the simple inheritance pattern that we would expect. The number's too big. The difference is too great. Okay. I'm going to come back. Oops. Here we go. Come back and check on everyone. Looks like we're okay. We'll keep cruising. Do another example problem. Okay, so in this example, we have cats where long hair is recessive to short hair, so short hair is dominant. Okay, and we have two heterozygous short hair cats mated. Since short hair is dominant, I'm going to use S's here. So we have two heterozygous cats, and I always put those little lines or those little hats over my lowercase s's to keep them straight from one another. And we'll fill out our Punnett square and then take a look at what our results mean. Okay. All right. Here we go. So right off the bat, what this Punnett square tells me is that 3 out of 4, or 75% of the offspring from this cross are expected to have short hair, whereas 25% are expected to have long hair. Okay. It does not matter. Remember, we're just talking phenotypes here. So it doesn't matter if the cat is homozygous dominant or heterozygous. It just matters if they're going to have short hair. Okay, so both of those would fall into the same category, hence the 75% as opposed to some other iteration of that. Okay, so possible phenotype outcomes. We just talked about this. You can be a short-haired cat or you can be a long-haired cat. So we have two possible phenotypes, which is again going to give us 2 minus 1 equals 1 degree of freedom. Anytime, for the most part, that you do a monohybrid cross, it's going to be 1 degree of freedom unless you have some sort of like incomplete dominance or co-dominance situation going on. Okay? All right. So we have that down. And next we're going to get our observed values. So we interviewed these cats. They also had a lot of babies. Specifically, oops, this is wrong. Let's change this. They had 42 short-haired babies and 8 long-haired babies. Okay, and we're going to see if that's statistically different enough from what we expected them to have. Okay, so if they had 50 babies, based on the results from the Punnett square, how many do we expect to have long hair and how many do we expect to have short hair? So take a second to do that math for yourself just to make sure you feel confident with it. Okay, remember we had 3 of 4 squares expected to be short and 1 of 4 expected to be long. So you can see what that would equal if we had 50 babies. Okay, I'm gonna go grab my calculator really quick in the other room. I want you guys to work on that math and I'll be right back. Okay, and I'm back. Hopefully you did the math. Okay, 3/4 of 50 is going to give me 37.5 babies. I know that seems weird. Don't round. I know you can't have half a cat, but in expected land, you can do whatever you want. So 37.5 is what we expect to have short hair based on our Punnett square, and therefore 12.5 is how many we would expect to have long hair, given the fact that we expect 25% of the offspring to be recessive and 75% of the offspring to be dominant. Okay? All right, so we're going to plug in all our numbers again. Null hypothesis remains the same. Results are due to chance. Any differences we see is just due to the random nature of mating. Okay, so we observed for long-haired cats, let me just look back, we observed 8. And we observed 42 short-haired cats. We expected, based on the numbers we just crunched, 12.5, okay, and 37.5. So I'm going to give you a second to crunch the numbers just to make sure that you can, okay, because you feel confident with this math. Remember, it's 3 steps: observed minus expected, okay, then you square it, then you divide by expected, and we'll meet back and I want to review just the final chi-squared value to make sure we're all on the same page and that you guys feel confident in your ability to do the math. So we'll meet back in like 60 seconds. That should be more than enough time to crunch some numbers. I'll make sure we have no questions. Oops. Okay, so after crunching the numbers, we should get 1.62 up here. Okay, and then we have 0.54 for short. Okay, given the fact that our expected value is so much larger and that's our denominator here. Okay, so then we should get 2.16 as our chi-squared value here. Okay, so chi-squared equals 2.16. Okay, our critical value is going to remain the same, but we can go back and look at the table just for fun. But we still have 1 degree of freedom. We're always going to use that p-value of 0.05, so 3.84 is what we're comparing to. We'll notice that, okay, our critical value of 3.84, this time our chi-squared value is not greater than 3.84. So the differences that we see are not large enough that we can say they're due to some other force. The differences that we see are in the range that they're still just due to chance and the random nature of mating. Okay, so because our chi-squared value is less than our critical value, we can either, you can say accept the null or fail to reject. And again, it really just depends on your teacher. On the AP exam, you can certainly say accept the null and get credit for it. But some teachers, especially if they've been teaching this for a long time, will still have in their brain that you can't say that and you can only say fail to reject. So I guess see what your teacher says and follow that because they're the ones grading you. Okay? All right. We'll probably do one more example here. We're going to do a dihybrid cross practice just because we started this last week and it's really good practice. So I'm going to zoom through this guy here and we'll use these results in order to do a chi-square that has a little bit more going on to it. Okay, so wolves are sometimes observed to have black coats and blue eyes. Assume that these traits are controlled by single locus genes and are located on different chromosomes. Assume further that normal coat color is dominant to black and brown eyes are dominant to blue. Suppose the alpha male and alpha female of a pack are black with blue eyes and normal colored with brown eyes, respectively. So we got dad, we got our male. He is recessive for both traits. He is black with blue eyes. Okay, so that's going to be lowercase n, why is this skipping on me? Lowercase n, lowercase b, lowercase b. And that's going to actually make for a really easy Punnett square. So if you were following along last week, the only gametes that this male can produce are lowercase n, lowercase b. Okay, lowercase n, lowercase b. Really, really easy then because our rows are all going to be identical because we're multiplying by the same thing. Okay, our female, on the other hand, is heterozygous for both traits. So our female is a little trickier. So she is uppercase n, uppercase b, lowercase b. If you're confused as to how to find the gametes here, I would suggest that you watch my podcast from last week. Okay, but you just use the FOIL method. Okay, so she can make 4 possible gametes. Yellow is a bad choice. She can make uppercase n, uppercase b, uppercase n, lowercase b, lowercase n, uppercase b, and lowercase n, lowercase b as gametes. Again, through the FOIL method, first, outer, inner, and last, you'll make sure that you hit all 4 possible eggs that this mama wolf can make. Okay? So the easy part here then is that these columns are all going to be identical to one another, given the fact that dad is given the same thing every single time, no matter what. Okay, so we're going to have some wolves that look like this, dominant for both traits. We're going to have 4 out of 16 dominant for both traits. We're going to have 4 out of 16 dominant for the first trait and recessive for the second. Okay. We'll have 4 out of 16 recessive for the first and dominant for the second. Okay. And then we'll have 4 out of 16 recessive for both. So this is a really interesting split. I think I alluded to it at the end of the podcast last week if you watched. But it's really cool when you have one parent that's heterozygous for both traits and one parent that's homozygous recessive for both traits, because you expect an even split between all 4 phenotypes. Which is not really common. So you would expect a 1-to-1 to 1-to-1 ratio between dominant, 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 recessive, recessive, dominant, and recessive, recessive, which is pretty cool. And we're going to use that information then as our expected value. All right, so possible phenotype outcomes. This time we have 4 possible phenotype outcomes, right? You can be normal coat brown eyes, normal coat blue eyes, black coat brown eyes, black coat blue eyes. Okay, so that gives us a fun and exciting 3 degrees of freedom this time, as opposed to the 1 that we've been seeing. Okay, so that's going to give us a different critical value then from our critical table. And then here are our observed values. Okay, you can see they're kind of all over the place. All right, so our expected values, if they have 100 babies, remember that we expect an even split, or we expect a quarter of each to be each phenotype. So we would expect 25 of each phenotype based on the Punnett square that we just did, the dihybrid cross. Okay, so our expected is going to be 25 down the line. Sometimes students get confused and always like to make expected the same number. Remember, that's only if that's what the Punnett square tells you to expect, right? If the Punnett square tells you that 75% of the offspring should be a certain way, then don't use the same number for all of them. So you got to think about that, okay? And our observed, 56, 20, 17, 56, 20, 17, oof, yikes, and 7. And I'm sure you can tell right off the bat that this null is going to be rejected because these numbers are vastly, vastly different from what we expected. Way more than is going to be allowed with our chi-squared values. So I'll give you a second to do these calculations, and then we'll meet back and review, and potentially, depending on what time is it, we might have time to at least start one more problem together. Calculations as well. All right, so hopefully you got a chi-square value of 54.96. That's massive. We know it's going to be bigger than whatever the critical value is, but just for the heck of it, because we have a different degree of freedom this time, let's take a quick look at what we're comparing to. 54.96. A whole bunch of pages. Okay, so we have 3 degrees of freedom now. So our new critical value is 7.82 as opposed to 3.84. You'll see with the more possibilities, there's actually a higher number. So because there's more chance that things are due to chance if there's more possible outcomes. 7.82 is what we're comparing to. This is obviously way greater than our critical value of 7.82, and therefore, when we have lots and lots of differences, we can go ahead and reject the null and say that these are not due to chance and that something else was going on in this wolf population. Reject the null. Okay, cool. There we have it. All right, let me just double check, make sure, whoops, that we don't have any questions, and then we'll move on to our very last practice problem. Okay, looks like we're good. This next one is interesting. It's about carrion beetles. And basically, carrion beetles lay their eggs in dead animals and then bury them in the ground until they hatch. How nice. So we're going to assume a few things. So preference for fresh meat is dominant to the preference for rotted meat in these beetles. Okay, so they prefer to bury their eggs in fresh meat, and they prefer to bury the meat shallow as opposed to burying it deep in the ground. Okay, so suppose that a female beetle is homozygous dominant for both traits, okay, and mates with an individual that's homozygous recessive for both traits, okay? So this is our P generation, and I'm highlighting this because it's going to get tricky in a second. Okay, that's our P generation. Then it says, what is the genotype of the F1 generation? Now remember, and if you don't watch my podcast from last week, F1 is going to be the cross between 2 P generations. So we can tell right off the bat, whenever you cross a homozygous dominant with a homozygous recessive, all of the offspring will be a mix. All of the offspring will be, therefore, heterozygous. Okay, so that's the genotype. The phenotype of the F1 generation is dominant for both traits. So it would be preference for fresh meat and shallow digging tendencies. Okay, so that's our F1 generation. Okay, the question here is what's going to happen then with our F2 generation if 2 of the F1s are mated. So if we cross 2 heterozygotes. Okay, and because this lesson is not about dihybrid crosses, but there is one from last week about that, I'm not going to do this whole dihybrid cross, knowing that, and hopefully if you watched, you know this too, when you cross 2 heterozygotes together, you always get a 9:3 to 3:1 ratio in your results. 9 dominant-dominant, 3 dominant recessive, 3 recessive dominant, and 1 recessive recessive out of a max or out of a total 16. Okay, so I'm actually going to skip this part, and we will expect 9 out of 16 to be dominant dominant, 3 out of 16 to be dominant recessive, 3 out of 16. And if you're like, how did she get this? You can do the cross yourself or watch the video from last week, because I go into lots of detail and I do one of these double heterozygous crosses. Okay, so we have these numbers, okay, and we need to figure out if we have 100 babies, right, what is our expected value for each of these? We have, hi Addie, we have dominant dominant, dominant recessive, recessive dominant, recessive recessive. So 9 out of 16, I'm going to do the math really quick. We would expect 56.25, and I'm going to keep that actually. You can, there's always, so the good news is with the answer for AP, there's always like a big range of numbers that you can give based on how you rounded it. So don't stress too much about how you're rounding. Okay, 3 out of 16, 18.75 for the next two. Okay, and again, all I'm doing here is multiplying the expected frequency from our Punnett square that we did not do, right? But this is a known ratio, and I'm multiplying that by 100 in order to see out of 100 beetles how many would be each of our expected phenotypes. Okay, so those are my expected values: 56.25, 18.75, 18.75, and 6.25. So when we go to calculate this then, 6.25, is it 18.75? Yeah. 18.75 and 6.25. You guys can go ahead and start crunching the numbers. And we'll see if the results that we see from our carrion beetle population are close enough to what we would expect or not. Okay. Okay, so you should get a number. Hopefully I didn't work too quickly there, but hopefully you're following along. Number is 1.89. Okay, so that's a pretty small value. So right off the bat, hopefully you're kind of thinking, okay, these results then are just due to chance because these differences are pretty close. These numbers are pretty close to what we expected. Okay, our critical value here, I'm not going to go back to the table because we did it in the last problem. It's going to be the same. Okay, our critical value is 7.82. Okay, given that we have, again, 3 degrees of freedom because we have 4 possible phenotypes. Okay, and 1.89 is a lot less than 7.82. So we're going to go ahead and accept the null and say everything is according to plan with these beetles. The results are similar enough to what we would expect them to be, and all is well. Okay. So that's our final example. Hopefully this provided some helpful insight if you've been struggling with chi-squared. If you haven't learned this yet, then it's a really good starting point for you, and hopefully you feel then when you do learn it a lot better about it. But it's a pretty can be a pretty challenging topic, but as long as you keep yourself accountable to thinking about what you're doing as you go, maybe remember some of those helpful hints, you'll be all set. So thank you for tuning in. We have non-Mendelian genetics next week, so bring a friend, and hopefully you found this informative. Have a great rest of your week, and I'll see you next week.